Hello everyone, thank you for joining and welcome to today's webinar session on validation of NGS in the HLA Laboratory. My name is Baron Victor and I'm the Product Manager for Next Generation Sequencing Products here at One Lambda. Today I'm happy to introduce you to Dr. Eric Weimer from the University of North Carolina Hospital at Chapel Hill HLA Laboratory. Dr. Weimer earned his PhD from Wake Forest University School of Medicine in 2009, followed by completing his postdoc at Duke University Medical Center studying regulatory B cells and infectious disease. In 2012, Dr. Weimer began his clinical immunology training at UNC Chapel Hill, where he remained on as faculty in 2014. His HLA laboratory began using NGS for typing applications in 2015 and has since performed several NGS validations subsequent to their initial one. Today, Dr. Weimer will be talking to us about the complex subject of validating NGS technologies in the HLA laboratory. Please note, this presentation will be recorded for future viewing through One Lambda's website. And during the presentation, please feel free to ask questions of the panelists by typing into the open field labeled Q&A on the bottom right-hand side of your screen. Your questions will be answered at the very end of the presentation. Also, please note that a short survey will be emailed to all webinar participants in approximately 5 to 7 business days following the conclusion of today's webinar. A total of 1 contact hour of ABHI-approved continuing education credits will be provided to those who complete the survey. It is now my distinct pleasure to turn today's program over to our presenter, Dr. Eric Weimer.
All right, thank you for that introduction. So today I'm going to talk to you about validation of HLA typing by next generation sequencing. This topic has come up numerous times over the years, and there's often been a lot of questions surrounding some practices and how to approach this topic from a number of different angles. So to start, the requisite disclaimer slide. What you'll hear about are essentially my views and opinions on this process and don't represent One Lambda's views at all, and I've received an honorarium from them for this presentation. So by overview, we're going to talk about the approach that UNC specifically took to validating next-generation sequencing. We'll talk about some of the summary data that we have in terms of how we approach the idea of QC in this complex process, as well as the separate issue of next-generation sequencing analysis that's done for HLA typing. And then towards the end of the presentation, we'll talk about some of the things that we've learned through our time of doing next-gen for HLA typing. It's been now approaching just about 3 years that we've been doing this, and so we've made a lot of mistakes along the way and learned a lot along the way, so we'll share our experience and things to consider in this process, as well as I'll talk about how best to smooth the process of submitting your ASHI submission packet to your commissioner for your accreditation for this process. So to start with background, I think many of you are probably already familiar with this. Sanger sequencing, traditional SBT method, you're sequencing two chromosomes concurrently. As many of you already know, this leads to a high level of ambiguities that need to be resolved. In our data, that actually represented that 53% of bone marrow cases had to have some form of additional testing to resolve these ambiguities. So that's a lot of additional work and cost and time put in to resolve all those ambiguities. Thus, the switch towards next-generation sequencing, or probably more appropriately called massive parallel sequencing, where because you're sequencing at a greater depth of coverage and you have the ability to sequence single molecules via this approach, you're able to phase across HLA genes, so that phasing ability allows you to assign particular base calls to specific HLA alleles, so you can know which single molecule they're on, which reduces the amount of ambiguities you have in your typing data. And then for most of the commercial assays that are available, they're approaching this from essentially a full gene perspective rather than a targeted just exon 2 or exon 2 and 3 and 4, for example, that often was done via Sanger sequencing. So we're going to talk a little bit about basic next-generation sequencing, so we're all on the same page. All assays kind of follow the same schematic. So you generate your PCR amplicons. Many of the assays use this as a long-range amplicon. There are approaches where you're essentially doing targeted amplicons, so not long-range. You're going to fragment that DNA so it'll work with your sequencing platform of choice. It has a number of ways in which you can undergo library preparation. And then typically that library, so a bunch of DNA molecules that are prepared for sequencing, are immobilized on some solid substrate. So this would be more of the ion torrent pathway here, and then this is kind of the Illumina chemistry here. You can perform some sort of clonal amplification, and then you're going to detect the incorporation of nucleotides throughout sequencing, and it's done by a variety of methods you can see listed there. So this is a general schematic that applies against the two dominant sequencing platforms that are available. So when you talk about different platforms, they all have different issues in terms of error rates. So you can see here, this is a table I've modified from 2015 publication where it has the ion torrent, the Illumina MySeq, PacBio, and the Oxford Nanopore MinION there, and you have their relative error rates that you see there, with the MySeq being the lowest, followed by the ion torrent, which are the two most commonly used, at least most often within a clinical lab space. So when it comes to data analysis, it's often two approaches. One is where you have reference-based alignment. That's meant to be highlighted there with that kind of blue color that's underlined. As you have your individual reads, they can be aligned to this sequence. Here, as so, and you can see in red, are various, would be single nucleotide polymorphisms, or SNPs, at those positions. And then you'll often, people refer to the depth of coverage, and I've already referred to it once so far, and that's essentially the number of times you've seen a particular base at that position. So I'm highlighting several G's at a location there, as well as several C's at a location more 3' on that sequence there. And essentially it's a sum of those sequences. The higher the number, the higher your confidence in that call typically is. And the second form is de novo assembly. So this is where, because they are barcoded and you know they came from a single molecule, here you can essentially tile them together to essentially generate your own brand new sequence, and that can be compared to an IMGT reference sequence to see which it is most likely to be consistent with. This has issues that aren't really pertinent for this discussion, but one of them is the value issue around accurately determining homopolymer stretches, which are prominent within the HLA genes. So in regards to ASHI NextGen accreditation, this is considered a new test system here. We'll have them send out these slides to you. That top one is actually linked to the ASHI validation checklist, which will bring up a PDF document that lists the necessary submission guidelines they would like you to have when you submit this to your commissioner for accreditation purposes. But in essence, it's 50 unique samples plus 20 blinded samples with a minimum concordance of 80% to your first and second fields. And then one of the considerations that people always ask about is where are you going to get 20 blinded samples, particularly for loci where ASHI or CAP or UCLA surveys maybe weren't going to cover, right? So DP alpha, DQ alpha, DP beta, for example, may or may not have been assessed on various sample types that you're wanting to, and there's various, we'll talk about various avenues for you to pursue in that for blinded samples. You want them to be samples that you routinely receive. I think that makes logical sense. Your runs should mimic what you have routinely had in terms of your clinical runs. There, you'll have to undertake precision studies, the inter and intra-assay precision studies. And as I mentioned, you can follow the document that there very easily lays out the necessary pieces of information your commissioner is going to look for when assessing your level in giving you accreditation for this process. My strong advice, and I'll mention it several times through this webinar, is collect all of the information that you generate. So this is going to be the lot numbers, the expiration dates. You're going to want to make some form of tracking for all of that as you would for any normal clinical assay. And at the end of this, you're going to gather all that information, and you're going to send it to your ASHI commissioner for review and approval. So in terms of the standards that apply to next-generation sequencing, all of the relevant molecular standards apply. So that's important to remember because there is a subsection for next-gen specific standards, and it's listed there as D5211 has this subsection of next-gen specific, and I've just shown some here that we'll go into greater depth and talk about later. So for our approach, initially we only wanted to utilize next-generation sequencing for just our bone marrow patient and donor population. So we focused on all of the alleles actually shown in bold are the ones that we routinely report for that patient population there. And then subsequent to this, our going live with next-generation sequencing, we moved to utilizing next-gen as our essentially dominant typing methodology for all patients that we receive. So then we started incorporating solid organ recipients and donors in this pathway. And as I mentioned, you want your run size to mimic your expected volume. So you don't want to, just because next-generation sequencing has the ability to multiplex many, many samples, 96 in many cases at one time, if you're routinely only going to be doing 6 samples at a time, that's more appropriate and representative of what you're going to be having. And that has impacts on the number of reads that every sample gets, and that then downstream detects whether or not you're going to have higher or lower amounts of contamination, for example, that you can cause issues with typing purposes. How are you going to QC every run? That's going to be a factor that you're really going to want to explain to your commissioner. So we utilize a previously typed sample on every single run for a couple of purposes. One, that it helps give us a QC parameter for the barcodes or indices that are used to identify individual samples, so we can ensure the fidelity of that process. Barcodes are moved around on known typed samples so that we eventually use every single barcode that we will use with that particular kit that we utilize. And then since you have some reagents, we utilize an Illumina MySeq platform here that are basically one-off reagents. You have the MySeq cartridge and the flow cells. Once they're used, you never get to use them again. So how do you go about QCing that process? You can get single lots of many of those reagents, which aids in that process. Also, having a previous type sample can benefit in many cases in that scenario. And then there's additional level of controls. So that kind of QCs the overall entire process, right? So that was extracted on the same platform, prepared via libraries, run on the same instrument, analyzed on the same software. And then you have other controls that can be utilized for assessing variability of instrument over time, the performance of your instrument over time. And those are things like the PHI-X that's available for Illumina sequencers, that's there. NIST has a reference material you see listed there that's available for purchase. CDC has a number of HLA typed samples that can be utilized for this sort of purpose and throughout. And also one of the things that has come up a number of times is whether or not the inclusion of a no template control to the process is beneficial. And that's something you're going to want to think about in your processes. There's pros and cons. We do not utilize a no template control through the entire process. We just do it as part of the PCR quality control. So for our timeline, you can see that this is covers almost a span of a year. So we started way back in November of 2014, essentially putting together our own set of commercial next-gen reagents to try to get this purpose going. And then early 2015, we moved to a commercial kit and really in thorough began doing our validation from there. It took until, August of 2015 to receive our ASHI accreditation. We've been live with it since around that time as well. So this is really meant to illustrate this will take some time, not only from the technical time of actually performing the validation, but to do it properly, and I think to really feel confident in the results that you have and to know your assay. It's going to take a lot of analysis time in terms of understanding where the issues and holes are with whatever reagents you choose to go forward with. So for our purposes, we took an in-depth look at how did sample concentration and HLA typing impact. So A is just simply representative gel electrophoresis, showing the amplification of each of the HLA loci with our particular reagents. So you can see their different sizes and banding patterns there. And then we looked at our main two specimen types, peripheral blood and buccal swab, and how well they performed for each of the various loci. And then we went on to do some further analysis based on whether or not we did additional purification of buccal swabs to increase their success rate, which is shown in E, that additional purification did. And then whether or not the buccal swabs, being the most problematic specimen type, could be improved solely based on having a higher concentration, and not surprisingly, the higher concentrations worked better. But I think the takeaway from all of this data that's shown here is that if you do an analysis and in-depth review of your specimen types and the issues that come up as you're undergoing this validation, it'll help you understand when you go live with it what to really kind of expect, whether are you going to normally see issues within DQ beta 1, or is mostly that's going to not be a problem, but you have issues within DP beta 1 for this particular specimen type. And that will help you troubleshoot and understand the limitations of your assay, which is really what this is all about as a process. So when you talk about specimen quality, obviously that I'm mostly referring to buccal swabs. So was it last year, we did an evaluation of looking at different kind of sample characteristics, whether it was total genomic DNA isolated, their level of fragmentation, which is being determined using a tape station, which gives you a DNA integrity number. It's essentially just how fragmented the DNA is. The lower the number, the higher the fragmentation. So a 1 is bad, a 10 is good. As well as, kind of standard sample quality parameters, the 260-280 or 260-230 ratios, and look at their sample characteristics on the success rate of HLA loci. And A, B, C, and D, you basically can see that the sample characteristic that most influences likelihood of successful HLA typing was how fragmented samples were. That's shown in B. Whereas the other parameters didn't really have a drastic change in some cases or an influence on how well samples were able to type. And so when you look out in E, we basically just broke out samples that had successful next-gen sequencing versus unsuccessful. They failed for a variety of reasons, and you can see that there really was this discrepancy between those two groups, so consistent with what we were suspecting based on our initial findings. And then when you do an ROC analysis, looking at all those parameters, the DNA fragmentation, which is the solid black line, came out to be the one most significantly associated, though it is clearly not the only factor. There is clearly a combination of factors that play a role in determining this role, but fragmentation certainly is one of those predictor of this. So this can be utilized as some form of quality control in this process, particularly for the buccal swabs or specimens that are known to have issues or fragmentation within them. So when you look at our kind of overall validation numbers, you can see the number. This is essentially quantifying how likely they were to PCR amplify at the time. And really the take home from this is you'll see most of the time they amplified without any issues. Most of these numbers are in the mid-90s. You'll notice that some of them are in the mid-70s for the PCR success there. And that's really, that DP alpha 1 is really meant to highlight that even though you can do a gel electrophoresis to see if you see PCR amplification, we've actually, on a number of cases, actually shown that the lack of having a band on gel electrophoresis does not indicate a sample will fail to get good sequencing data. And that's really due to the sensitivity of next-generation sequencing to pick up low-level amounts of DNA compared to that of electrophoresis. And on the bottom table is a summary of the average Q30, so the quality of those particular reads for each of those loci, as well as the average coverage for each of those loci, as well as potential novel alleles that we saw there. And we'll talk about that as a subcategory. And you can see most of these are very, very high. Genes are covered at a relatively high depth for our purposes. So there was really no issues here. So when you kind of look at the stats of our validation, we had a little over 3,000 total alleles. 212 of those were unique. There were 6 that we got incorrect. I'm covering up a box that's not relevant anymore. It has to do with ambiguities that we found in this assay, but the assays have such been improved upon where that's no longer a valid concern. You can see the total number of samples we ran, number that were unique, and then our precision studies. And you can see all of the analyses there. It came out basically to be highly accurate. The precision was 100%, and our ambiguity rate was about 3%, which is much lower now, given that the assays from all of the vendors have been continually improved and have addressed some of the issues that were initially present in many of the available assays. So that number has come down drastically. So then the big question that everybody wants to know, and I think is really important for you to understand when you are preparing your validation, is why did we miss these 6? To investigate, in my opinion, they're the most valuable of all of the samples we ran, of all the loci we even looked at, really. Because they actually tell us issues with our process, how we were doing analysis, issues that are associated with the assay that we may need to address to better address going forward. So, you know, when we did our investigation, these were mostly due to some form of contamination and the occasional allele dropout. And that was not surprising, considering that we were early in this process, and as we were first doing our validation, you know, you're unsure of how exactly to do library preparation. You don't have the confidence you'll have later on. So, you know, we made small errors. We learned things about the assay and how better to utilize it and handle it to improve this issue. And then the next question is, okay, well, now you know why you're having these issues, but what are you going to do about those assays? How are you going to handle that going forward so you don't make mistakes on clinical specimens? And that's the crux of what we're talking about. That's what you're going to need to explain to your ASHI commissioner so they feel confident in your ability going forward. So we developed a set of criteria that we really adhere to. So they mostly revolve around depth of coverage and the quality scores. So this is the graphical representation of that table that I showed you about, and you see in gray for B and C. That is where our defined criteria were at that particular time. We've, as part of our normal quality improvement processes, have continued to monitor these over time and adjusted accordingly. So we actually no longer continue to use the Q30 score as a valuable metric for determining a successful run, because we found that over the course of a year and a half or 2 years, that parameter actually never failed, thus was not a very good parameter for us to continue following. So coverage ends up being the one that we found is most important to this particular analysis and one that we continue to use to this day. And then the second tier to this is purely instrument performance and assurance that our sequencer is operating normally as we would expect it to. So in A, you have the cluster density we saw during our validation. B are the clusters passing filter. C is the read quality. And then D is a measure of the error rate using the phi-x control that we utilize there. And the point to make is that, so we trend these over time so we can make sure the instrument is not having any issues. And one of the things I find quite interesting is if you look at D, where initially in the first like almost 7 runs, you see a relatively large swing in variation, and then somewhere around run 15-ish, it almost levels off and bounces around that level, and that's because at that time we had a number of runs under our belt, right? And we became more confident in our ability in the assay, and the error rate essentially leveled out there. And so we utilize those parameters to this day to track instrument performance and how well it's working. And as I mentioned, the rotation of barcodes for fidelity purposes of index primers. So here's just kind of a general look at the specific quality metrics that can be used from one of our policies of whether or not you have specific read quality measurements, how much phasing in terms of analysis are you going to allow, depth of coverage, whatnot there, and various parameters that you want to look at to consider a successful run. And I only make the point at the bottom where we're setting the depth of coverage criteria at 70, and from our analysis we can actually say that we've gotten accurate HLA typing down to a depth of coverage of 50, and I actually think now we're gotten it around 34 depth of coverage and still had successful typing. But to be conservative, we've left it at 70, and this really hasn't been an issue in terms of repeating runs, for example. So when you look at analysis, it's a separate validation of the software. So if you're using a MySeq or other platforms, they have multiple components. There's a MySeq controller and a MySeq reporter software that can be updated and will have to be validated as would any other software system. And then the point really that I want to drive home is the just general vendor software accuracy is actually quite high. In our hands, with no user intervention, just purely loading the sample into the vendor software, letting it do the analysis, and give me back an HLA type. There was a 99.7% accuracy in that process alone. Now, it's not 100, but the ones where the software was incorrect was always flagged. So all of the software that the vendors provide have various mechanisms to identify issues in the sequence, whether it be homozygous, whether it be linkage disequilibrium isn't present, is it a rare allele, a novel allele, for example. All of these things are flagged in various ways and metrics by the vendor software. So if you follow those, it's been very successful in our hands. And then when we first started, this was a new process, and so if there was any question what the HLA type was, we would repeat it using, at the time, Sanger sequencing, and now that we've discontinued Sanger sequencing, other technology has proven in some cases to be beneficial, and it really just depends on what your issue is for that particular HLA loci, for example. And then, are you going to confirm rare alleles? Well, how are you going to help address the transition from kind of the workflow your staff has been used to for many years in cases of Sanger sequencing to now this high throughput multiplexed pathway of next-gen sequencing, and make sure that they feel comfortable throughout this process? And I think if you do this sort of analysis that I've been talking about, it really increases their level of comfort, as well as if they can say, okay, well, I repeated this sample that I was a little unsure of for these handful of reasons. I repeated it on a different technology and I got the same answer. Now I feel confident about it. And so we did that for a relatively short period of time, because it won't take long for them to gain confidence in this process, and then we essentially only do next-gen sequencing now. So, we're all held to different turnaround times based on our transplant centers. So here I'm showing for us, way back when we were doing Sanger sequencing, we had the very unfortunate of having a turnaround time approaching about 21 days or so using Sanger sequencing because of all of the ambiguities that needed to be resolved. And then when we switched to the first version of the assay that we utilized, that dropped it down about 7 days or so. And then, assay improvements, our own improvement with the assay and workflow enhancements, such as not waiting to maximize run efficiency in batching samples. We would do set run schedules, reduced it even further. So from about 17 days, dropped it down to about 7-ish days, and where we are currently to this day. And that's a testament to really the commercial vendors and their continual improvement of their assays, where they've evolved, improved primers, for example, pooling strategies, for example, and then software has continually gotten better at detecting heterozygous alleles, so you don't get as many allele dropouts, and then the increased identification of the common, well-documented alleles. So if you apply this to the 1 Lambda next-gen product, I'm not going to show any data on this, but so for highlight purposes, they have the HLA genes shown in the table below are amplified in a single tube. The library prep process follows a very similar process that I talked about earlier. That's why I'm really highlighting the amplification in a single tube aspect. And this amplification process takes about 2.5-ish hours, and you can see the regions that are targeted there. So if you're utilizing this sort of pathway for your approach, what are the things that I would want to consider in this process? And one of those is, since everything is occurring in a single tube, the issue there is, you can't really go back and say, okay, well, did I have amplification of my DQ beta 1, for example, even though I got really bad sequencing data from that. Now, we haven't had that scenario, but using their assay. But if you have had it, then how are you going to help troubleshoot that process? And one of the things that we've done is look for amplification there. It's not 100%, as I talked about earlier. The other issues that I can think of having kind of a single tube amplification is, do you get preferential amplification of particular loci? I'll say in our hands thus far, that hasn't been really that much of an issue. And then along those same lines is the allele balance. So, you're supposed to have roughly a 50-50 balance of each of your chromosomes. Is there in the ballpark of that for the HLA loci that you're examining here, and what is acceptable to you? What are you going to use as a parameter going forward? And that's where running enough samples to really have a better understanding of what it is in your hands, rather than solely relying on what the vendors have told you, is extremely beneficial. And then for data analysis, I think one of the strengths is that, they have the ability, 1 Lambda has the ability to combine their next-gen sequencing data with their single-engine bead profiling data, and that many of us use for that purpose. So that's certainly, I think, a pro in their favor. And then, some of the more practical things to consider are how well and easy is the software to use. Is it just so overwhelming with the amount of information that's presented that you don't feel comfortable looking at it and it's just too overwhelming? Novel alleles are going to be an issue for you. They have been an issue for all of us that do next-gen sequencing. How well does the software identify them? Does it give you information relatively easy about them? Are there amino acid changes? Is there a stop codon? Where is it located in the gene, for example? Now, if you're going to use next-gen for solid organ purposes, which many labs do, does the software have the ability to convert the high-resolution type you're getting to a serologic equivalent, which I will tell you firsthand is of extreme benefit to the lab. I'll tell you that their software has that ability. And do the software appropriately flag any issues that are present? As I mentioned, they're often very accurate in and of themselves, but when it's flagging you, is it appropriate? Does it have issues that you feel are necessary? And then along those same lines, is it doing it too often, right? Because you can have a software that flags you for various issues, but then if your protocol and setup is going to be that you only look at those that are flagged, is it too frequent for you to feel comfortable with? And then I think it's important to understand how accurate the software is with absolutely no human intervention, because that will help you, I think, understand where issues are in the analysis pipeline for your assay. So when you talk about our lessons that we've learned over these years, one of them is how are you going to store the data? Each run typically generates about 8 gigabytes of total data, which isn't a super overwhelming amount, but as you consider the growth over time and having to maintain files, that can get onersome over time. So how are you going to handle that? That's something you'll have to address in the package you provide, because you see I listed there the ASHI standard about the files and what your data storage policy will be, where, when, how sort of approach. And really the biggest take-home from this altogether is to know the assay you choose to go forward with. What are its holes? What are its limitations? Does it have issues with homozygous samples? Because it is very sensitive to detecting a heterozygous, so even if the ratio is 95-5, it's still calling heterozygous samples, but that seems too sensitive for you. Then you need to understand those limitations so you can better policy approach those samples going forward. And then essentially, what is going to be your limit, right? Where is your limit for allele imbalance? We accept a 90-10 ratio as our biggest allele imbalance. Anything greater than that, we feel the software gets too questionable to call. What's your line going to be? We do check our associations. When we had allele dropout, way back on those slides when I told you there was some dropout in our initial validation, simply checking the known DR, DQ, B, and C associations really caught all of them. There was one scenario where that didn't catch it, and we utilized some different other approaches to aid in that interpretation, as well as identifying any potential contamination, which is mostly in your DR beta 3, 4, and 5, for example. And then you're going to have to understand the process for novel alleles. What's the HLA allele nomenclature? If there's mutations in exon 5 of class 1, how does that impact how the naming of that, the proper naming of that would be? How are you going to handle it? Because you will have them on run number 1, you do, when you go live. We've all experienced it. And how are you going to report them as for your clinical colleagues, interpretive, for their match runs and patient understanding? How are you going to report them to them? Communicating those results becomes critically important. So I want to touch on one other aspect, and that since within next-gen sequencing, you're doing so many multiplex samples. So you have all these different samples with their various targets, whether it's HLA A, B, C, and all of them end up getting pooled together, right? So then when you do your sequencing, you demultiplex that out and put all of the right sequencing data with the right sample. Now, one of the things that we have experienced in this time is it is incredibly valuable to evaluate that process and understand how repooling of those samples. So you load a run onto your sequencer. The sequencer has power failure. It fails to run. It has debris, and it doesn't function appropriately. Now you've lost that sequencing run, or it's not high enough quality that you feel confident in it, and you want to redo it. You don't really want to have to go all the way back to the beginning of PCR to redo everything. One, that costs a lot more money. Two, that delays the results for those patients. So evaluating how to repool samples can kind of get you around that process. And we've utilized this a number of times, particularly for samples that have been troubling or loci that have been troubling in a sequencing run. And then, this has been a question that comes up a lot of, you'll have to make the decision of, do you want to maximize the cost effectiveness, essentially holding samples to maximize run throughput, or are you going to really kind of look at getting the lowest turnaround time you can for them? And that's individual lab, transplant center level decision to make. Many of these processes lend themselves very well to liquid handlers or automation. And then if you're going to take that approach, which is very beneficial for this kind of hands-on library preparation approach, you're going to want to validate not only that process, but you also have to undergo traditional carryover studies to look at contamination of those. Does contamination get carried over from various wells? And then remember that the ASHI has standards regarding pre- and post-amplification areas. So the way as it currently stands, you have to decide which steps in the process you want to use your liquid handling robot, unless you're fortunate enough to have multiple. And then I always make this point on the bottom about how will you change how you bill patients. So the CPT codes for HLA typing are not based on technology. We have some of our colleagues in other laboratories that is the case. So there is no obligation to change how you bill patients. You can use the exact same CPT code you were using before. It will be cheaper, so you can make more money in that aspect. If you choose to, you can reduce the cost to patients if that's what your institution chooses to do. But this is really to point out that CPT codes for this process don't actually change. So to finish really talking about the accreditation process, when you submit this packet to your commissioner, you're going to have to also include the policies around staff training. How are you going to address staff competency, staff training? Who's going to do it? How are they going to do it in that process? We treat them as two separate entities, the technical component of performing it, and then the analysis component are two separate training aspects at UNC. How and who will do it? Does everyone do it? Are you only going to have a technologist followed by a supervisor or a director? What's your pathway going to be? They're going to want to see that laid out for them to evaluate. And then as we've talked about numerous times, what criteria for analysis are you going to utilize for determining a successful run, and what are you going to consider those to be and why are the important questions to kind of answer. And then you're going to want to take all of those worksheets you've been gathering through all of your validation runs and have them as part of your submission packet. It'll help show your commissioner that this is what you're going to do tracking-wise. This is how you can identify if you have expired reagents. Many of these kits have many different boxes and components and tubes associated with them, so it's fairly complex. And how are you going to ensure that those are QC'd properly and that the reagents are being used prior to their expiration dates? All this obviously is a new procedure and policies that have to be reviewed and signed off by the lab director. And then I actually think it's important for you to provide some form of rationale for whatever it is your laboratory chooses to do. How are you going to prevent false negative, allele dropout, or false positive, say a contamination of DR beta 3, 4, or 5 results from going out? How are you identifying them and preventing those results from reaching patients? And then what are the limits of your system? This validation, as it is with any technology, it's really to figure out where the limits are. And for you to understand that, you have to run a number of samples. Our estimation is about 100. Someone usually asks that question. Based on the amount of allele dropout or issues you've seen at roughly about a 1% rate now. So if you run 100 samples, you're potentially likely to see one specimen, right? So understanding the limits of your system. So again, I would follow the instructions that are set out as the validation summary for new systems, and that document is available on the ASHI website. Think about your processes and where is next generation sequencing really going to fit in. Do you want to use it for all of your typing, for if you're a bone marrow and solid organ transplant? If you support both of those programs, are you going to use it for both or just one? And then kind of plan out your overall strategy so that way you have all of your new procedures, you have all of your policies. We met as a laboratory probably at least 6 times prior to going live to talk about what our policies and procedures would be, how we would handle various things. The staff could ask all the questions they had of the directors to get their questions answered about how and what and where and when. We would do various things, and then I cannot stress enough the inclusion of your IT department in this process so you can have some form of IT infrastructure available to you to store data, to help with analysis if that's what you're going to use as like a server-based analysis, as well as laboratory information systems. And once you have all of that, you can simply compile it all. I would do it as neatly as possible and send that to your ASHI commissioner. And how the blinded samples are done, that representative will provide those reference typings to your commissioner for comparison. And then I really think it's important that you keep a log of samples that have issues, not only because that's good laboratory practices, but it also, those are the most informative ones, as I talked about. If you have a samples that are routinely having issues, and you can look at that and you can say, oh, well, they're almost always peripheral bloods with a DNA concentration of 600 nanograms per microliter, well, then maybe something's going on with your DNA extraction or those particular samples that you want to investigate so you can better understand the process and maybe not put as many samples through that have issues. So I'm going to end there, and it's really important for me to acknowledge the individuals who do all of this work. So not shown in this picture is Maureen Montgomery, who's been fantastic and did a lot of the experiments that you saw, particularly for the buccal swab data that I showed you. And I really want to tell you that in this picture that's shown, which is everyone in the lab, here, 4 of those people in this photograph don't routinely perform laboratory testing. So there are 6 individuals in our laboratory that basically do all of the laboratory testing that goes on here, and we've been able to do that. It's a testament to their work ethic that they get so much done efficiently as they do. Well, nice to hear that. Thank you very much, and I'm glad you were able to listen in.
I guess that's probably where we're going to stop. So again, thank you again very much, Dr. Weimer, for delivering such a well thought out and detailed presentation. And thanks to everyone who was able to join us today. We do hope that you found this webinar presentation informative.